So this video is an introduction to the Estimating Population Size and Density Mark and Recapture online simulation. So in order to analyze population growth of any invasive species or any local species, or to investigate carrying capacities, ecologists have to have a way to estimate population sizes and population densities. There are a lot of factors to consider when doing that. First of all, ecologists have to try to not harm the organisms or disrupt the ecosystem. It's very hard to estimate animals' population size because they're mobile, they move around a lot. And some animals really like to hide from us. And other species are very difficult to distinguish from one another. So when ecologists are trying to estimate plant population size, they randomly sample areas using what's called a quadrat. It is a square frame that they place on the ground and count the number of plants of different species inside that quadrat to obtain a sample count or a density estimate. If they were going to count larger trees, or larger plants like trees, for example, they do what's called a line transect. So they mark a straight line with a long piece of string and count all the trees within that distance that are that species. So for example, all the white oak. Now, because animals are mobile, when ecologists are sampling animal populations, they often use a procedure called mark and recapture technique. And in this procedure, biologists capture the animals alive, mark them, and then return them unharmed to their environment. And later, the sampling process is repeated. Scientists capture, mark, and then return the animals to their environment, and then recapture them. And there is a mathematical formula that we will be coming back to in a few minutes that I'll explain. But first I want to take you to this particular website where we're going to do a simulation and show you how to do the simulation. You will be given this link and you'll be doing this on your own. But if we go to this website and we hit go and then we hit open, if you look at the timer, it will start timing, and when we get to 2, we want to hit close. And if it's 2.01, that's close enough. Now this is the first time we captured voles. If you look, the ones that are in the blocks can't get out. So there's no voles in trap number 1, there's 1 vole in trap number 2. These guys are moving around and it's hard to count, and I think there's 1 in this one. So what we would do is we would, the very first time, mark our voles. So we have 7 total that are marked. So then we go to our lab sheet and we type the number 7 here. Now of course I'm going to want you to use red font because I like when the color is easier for me to read, and it's also nice if it's bold. Now we're going to go back to this website and we're going to release these voles back into the environment. And we're going to trap the voles 10 times, and we're going to see how many voles that we get back. If you get a lot of the same voles that you have already previously marked back, that means your population is pretty small because there aren't that many voles and you keep getting the same voles back in your traps. If you don't get very many of the voles back and you keep getting new voles each time, then you know you have a very large population, because statistically speaking there's so many voles you're not recapturing the same original voles. And by the way, I didn't mention this before, a vole is a little bit like a field mouse. It's a small rodent. So we're going to release the voles and then we're going to open the traps and it starts timing again. And you get to 2 and you hit close. So now we are going to look to see how many are totaled in our traps and how many are marked. So we are not marking our voles again. Notice we only marked them the first time. So if we looked here, the 7 total marked, that didn't change. That's the number you marked to begin with. The number that are marked that are in the traps is 0. The total in the traps is 5. So we go to our file and we go to number captured, and the answer there was, it was 5. The number recaptured with the mark was 0. And we're going to do that again. I'm going to show you again one more time. We're going to do it a total of 10 times, but I'm going to do it once more with you guys again. Notice I didn't mark them again. I'm going to release the voles, and then I'm going to open. Watch the timer until it gets to 2 hours. Thank God for time lapse. And if we look again, we have 2 that are marked in the traps and 11 total in the traps, right there and there. So we go back to our data table and we say 11 total and 2. Now let me explain how to do this calculation and this mathematical calculation. What you would do is you would do this 10 times total, not including the first time where you just marked the voles. So you have to mark the voles and do it 10 more times. And you get these totals here at the bottom. Now, it is important to note that it's not always going to be 7 when you do this at home. Every time somebody does it, it's a different number, and all your numbers are going to be different. So don't expect your numbers and your friends' numbers to match, okay? So when we do this calculation to give our population estimate, we take the total number captured, so that would be the number from here. It goes right there. And then we multiply that by the number marked, which in my case would be 7. That's the number from, the number marked was the number from here, this number 7. So you can actually plug that into your formula from the very beginning. So that's like 7 right here. And we could make that red and bold. Now this number is the total number captured. Like I said, that's the total from here. And then you have your total number recaptured with the mark. That's going to be a total from this column. So this total number captured goes right here, and the number recaptured with the mark goes down here. And then we multiply the total number captured by the number marked, which in my case would have been 7, divided by the total number recaptured. And then that is our population estimate. So then you're going to answer a bunch of questions. You're going to answer questions 2 and 3 on this paper. It's going to be a Google Doc, and you're going to submit it. And that's going to be worth a small number of points. There is an analysis section to this lab, and it's going to be under the quiz section in my MCPS classroom. And a couple things that I wanted to talk about before I have you do that is some vocabulary and some examples I wanted to talk about. So one of the questions asks about population distribution. And sometimes students don't understand what population distribution really means. It's not how many of an organism are in an area, it's how evenly distributed they are. So they're evenly distributed if you have roughly the same number anywhere you go in that ecosystem. But they might have uneven distribution if you have clumps where there's lots of voles and other areas where you're not going to find many voles. I also wanted to talk about sampling effort, because one of the questions asks you how you could change sampling effort for this particular activity. Now, the best way for me to explain what sampling effort is to give you an example rather than try and define it. And I'm going to use a different species as an example because I don't want to give away the answer. So when we ask how to change sampling effort, oftentimes students will tell me, oh, if you work harder and you'll get better data because you're putting more effort into your sample. It's not quite as simple as that, and that's not going to be the kind of answer I'm looking for. I'm going to be looking for specific examples. So I'm going to give you an example of what a good answer would be if I was talking about sampling effort with trees. So for example, if we were doing an experiment on plants instead of voles and we were counting trees, remember we talked about if you were going to estimate the population of trees, you take a straight line and a long piece of string and count how many trees that you hit in that distance. So for example, you could use a string that is 1 mile long, or you could use a string that is 2, 3, 4, or 5 miles long. Obviously, the longer string would increase your sampling effort. You also could do a string, a 1 mile string in 4 different directions, or you could do a 1 mile length of string in 10 different directions. So that would be some example of changing your sampling effort. Obviously, if you went in a greater number of different directions from that, you kind of put like a stake in the soil and, go in 4 directions versus 10 directions, it's greater sampling effort to go in 10 directions. So of course you want to have enough sampling effort to get good data, but you also don't want to waste time or money in your experiment. So hopefully you'll be able to answer the sampling effort question with regards to this specific experiment that we're doing. So how could you change the sampling effort if you were capturing voles?